Welcome to Barry's In the Interim podcast, where we explore the cutting edge of innovative clinical trial design for the pharmaceutical and medical industries, and so much more. Let's dive in.
Welcome, everybody, back to In the Interim. I'm your host, Scott Barry, and my usual partner in statistics here. I was going to say partner in crime, but that sounds bad, Kurt.
Does.
Partner in statistics, Kurt Veely, is with me today. So today, yesterday was before, tomorrow's after. We may always consider before the FDA draft guidance on Bayesian statistics, before and after. So Kurt and I are here to talk about the very much anticipated draft guidance. It's draft review comments, but the FDA put out a guidance document, the use of Bayesian methodology in clinical trials of drug and biological products, guidance for industry. And that is recently out, so we're going to talk about the guidance. By the way, interestingly, in that comment, it says drugs and biologics. CDRH, the Center for Devices, has had a Bayesian guidance out for many years. And so this is now drugs and biologics with the guidance. And I think if you're a listener to this program, if you've done many interim analyses with us, you know that we are Bayesian. We think Bayesian. We do frequentist trials, but we think Bayesian. We think there are many cases where they're really good techniques. And so we're excited to have this document out, and we're going to provide our reactions to the document. We're going to talk about pieces of it. So, Kurt, your first read, what is your reaction to the Bayesian guidance?
So, as a Bayesian, I think most Bayesians are celebrating this. I think all Bayesians are celebrating this. This has been something that we've been working on you've been working on it for 20, 30 years. So I've been working on it less, but certainly decades. And so it's good to see all of the things that we've been working on, all the different designs that have been developed over the last 15 years. This is really a clear message from the FDA that these designs are acceptable. It goes through how to do them rigorously, how to maintain scientific standards while these are happening. It goes through lots of examples on here are cases where we think we should use Bayesian. So all of this is great. And I think it's really important for the FDA to say with a clear voice, we will take Bayesian designs. We have taken Bayesian designs, and let's go. Let's do more.
Yeah, I had a similar reaction to it. Maybe I had a prior expectation for what the document was going to be. And I find it very, very positive. I think it's very well done. I think it's thorough. I think, and I'll contrast it a little bit. You and I talked about, and you can go back and listen to the podcast where we talk about the ICH E20 on adaptive designs that came out. It exists, and it's a positive thing, the ICH E20. But there's almost this, be careful what you're doing, almost this negative. If you really, really want to do it, here's the way to go about doing it. I was almost sort of pessimistic in the way it phrases things, overly concerning. This document, I don't think has that. I think it's optimistic. This is a good, positive way to go forward. I think it's appropriately providing guidance on cases in which you can do it. So I think it's a really good document. I think it's a very beneficial guidance.
I agree with all that.
Okay. So the various pieces to it. First of all, when it came out, the FDA commissioner, Marty Makary, Dr. Marty Makary, had a few quotes about it. So this was thought to be important enough that the FDA commissioner provided quotes. I found a couple of those. I think there's a short video that he actually came out with, which is really awesome. But Bayesian methodologies help address two of the biggest problems of drug development: high costs and long timelines. Providing clarity around modern statistical methods will help sponsors bring more cures and meaningful treatments to patients faster and more affordably. So, of course, very high level. This is good for patients, good for sponsors, but important enough that the FDA commissioner makes a comment about what some may think of as a statistical guidance, which is also a very positive thing.
Well, I think one of the key issues here is just how much, we've spent a lot of time synthesizing data. Just the world's just awash in data floating around, historical studies. We see all these meta-analyses. We see, the things that the guidance talks about, adult patients, pediatric patients. Synthesizing information and not reinventing the wheel is just a key part of what we do every day. And so this really, at some level, what this guidance is about is how do we synthesize information from multiple sources? How do we put things together? And how do we do that in a rigorous way? And so we would expect to see advantages to both patients and sponsors when that happens.
Yep. So both of us are very positive on this. I think it's a great document. Let's talk about what's in there, Kurt. They make a strong distinction, which is kind of interesting. Very early on in the document, they make a strong distinction between trials where there's a claim of type 1 error control and trials where there's not. And trials where they're not are tied to scenarios where external data is being brought in as part of it. Bayesian is being used to synthesize external data. There's information, and it very clearly states in those scenarios, type 1 error control is, I'll say, sort of awkward or less relevant. And the early distinction between those two cases of Bayesian, which, by the way, they're both reasonable cases where Bayesian could be used, where you're still controlling type 1 error, and those where external data are brought in. I thought it was really interesting that that distinction is made very, very early.
Well, I think, there's a history here on how we got here. We've done a lot of adaptive designs over the years. We've driven those by Bayesian aspects. In many cases, you don't have information coming in. You're doing lots of interim analyses. You don't want to make false decisions. We do want to put only good drugs on the market. We want to fail bad drugs quickly. The type 1 error control has been, a standard part of drug development for years and years and years. And so when you start talking about external data, this has been, if we talk about the last decade, it's been a struggle to try to put external data into the same box where there's type 1 error control. Because, and I think the guidance, the new guidance says this up front, if you already have evidence that a treatment works, then, you're not necessarily, you don't need type 1 error control for your new trial. You've already partially disproved the null. And the current trial is to get you the rest of the way there, to have the totality of the evidence be sufficient rather than just relying on this single trial. And so, we've spent a lot of time with FDA and other regulators, trying to say, well, here's what it does to the type 1 error. Is this enough? And it's always been an awkward conversation. And one thing this document's clearly hinting at where to go is let's just move into the brave new world where we can just synthesize the evidence directly. And I think that's a huge step forward.
Yeah, so one of the quotes from the guidance, and this is related to what Kurt brought up about if we're bringing in external data, it says, by borrowing information, one is assuming that the borrowed information is relevant, evaluating the degree of borrowing based on the expected outcome when there's no effect under the null, is philosophically inconsistent given a prior which assumes a non-zero effect. So the whole point is, how much do we care about what happens under the null if we've got evidence that says the null is unlikely? And here's a simple example. Suppose a company has run a phase 3 trial, rejected the null and has strong evidence and is running another trial. What does it mean to think about type 1 error in that circumstance when the probability of the null is already very low? And so that's the notion. And from a pure statistical standpoint, if you're bringing in external data that is positive and that data is used, you generally are not controlling type 1 error. Under this new trial, if the null is true, you can get this inflation. And it very strongly says, we're going to calculate that, but it's not necessarily constrained. And these are really the two separate cases of Bayesian.
Yep. I think it also gets at just what you need to do for the rigor there. There's lots of discussions, I think over the past, I don't know how many of these borrowing trials we've done over the past decade, but it's a lot. And, originally our discussions, we talked about the methodology. More and more of the discussions today get into, are we comfortable with what data you're borrowing? And that's a key part of the guidance here. Essentially says, if we're going to allow you to inflate type 1 error or discard the concept itself of type 1 error, you need to be bringing us real information that has validity, patient-level data, make sure your covariates make sense. All of that is in there, and that's been more and more a key part of our discussions. And I think you talked about it being a statistical guidance. It's also a bit more than that in terms of just a scientific guidance over, if we're going to synthesize information, what is information? What's relevant and what's not?
Yeah, they talk a lot about, and I think it's a case that our listeners can understand. It's hard to refute such a thing. They talk a lot about pediatrics and adults. One of the more common ways in which Bayesian borrowing has been used is to estimate an effect within pediatric populations using results from adults. So circumstances, there are a number of scenarios and diseases that are much more common in adults than they are in pediatrics. A simple example of this, and it's come up, is acute ischemic stroke. I was unaware that actually adolescents and pediatrics can have an ischemic stroke. They're very, very rare. So adults, it's a very, very common scenario, a huge burden that many therapies are working on. But we have treatments that are effective in adults. And now you might have tens of patients in pediatrics. This happens in a number of things that are quite rare in pediatrics. And we want to estimate the effect in that group. We can't enroll a trial of 600 pediatrics where we enrolled 600 adults. We have information about efficacy. We still want to collect the data. We want to run the trials, but we might want to borrow. And really, this is a statistical way in which the effect in pediatrics can be estimated by using the information in adults. And this is used in a number of examples. It's been used in a number of FDA examples already, and they highlight this as a case of borrowing. A hugely valuable role of Bayesian, and now they extend it. I found it really wonderful that they spent a good bit of time talking about the role of estimating the effect in one patient group, subgroups of related diseases. I think we're very much in the future and the present where we're looking at a particular indication and there are related indications. It can be severity of disease, it can be genetic classifications of the disease, slight differences of the disease, but they're related. and you demonstrate an effect or you have data in one, it's relevant to these other groups. And they spent time on this and they opened this up as very much a place in which Bayesian modeling can be used. I thought that was fantastic that they highlight that scenario.
Yeah, and they highlighted it under, I think, a couple different situations. They're related, but what we've worked in, one of these is basket trials where you may have a targeted therapy. We've worked on the ROAR trial that was for BRAF positive patients. And in that, the groups of patients in the trial are where the tumor is in the body. Is it in ovarian? Is it thyroid? Is it lung? Is it at various locations that the case may be? And, those trials, we got a pan-tumor. We, of course, the sponsor. I like to view us as part of the team always. But a pan-tumor approval. So essentially, you can treat any BRAF patient with this drug because of that borrowing between those groups that they were viewed as related. We've also done trials where the FDA is interested in sensitivity analyses, where you've got, say, more standard subgroups, severity of disease, race, sex, whatever the case may be, where we've said, this group, it only has 12 patients, but the result is a little bad. Should we worry about that? Or should we view this as it's more in line and it's just noise? And we've done analyses in there. It's actually the same models, but the FDA does distinguish them as slightly different questions. And I think that's valuable. That will, especially as we partition diseases more and more, how we do this.
Yep. They highlight one of the CID projects, which was for seizures, and there are different classifications of patients that have these seizures, and they're related. And they had run trials and had approval in 3 subtypes of this disease, and they were now looking at a 4th subtype. And they had very homogeneous estimates of efficacy, I believe 25 to 30% reduction in seizure frequency in these 3. And now they're running a new trial in a new population. And really, it's scientifically, should they have to run a 2.5% alpha controlled trial for every one of these new groups, or some level of barring? Very similar to the basket trial you talked about is another circumstance. So this I think it's a great use of Bayesian methods moving forward, and I think they'll be more commonly used.
Yeah.
So the other side of this are Bayesian trials where you're controlling type 1 error. And they make a comment in here, which I think is positive. They don't spend a ton of time on adaptive trials, but they do say that when you're calibrating type 1 error and you're making a claim that the trial has, say, 2.5% one-sided type 1 error, such an approach may be appropriate for designs where Bayesian approaches are used not to synthesize multiple information sources, but instead to facilitate complex adaptive designs.
So I think this is actually it's been an undercurrent of a lot of guidances, and why a lot of times people ask us, well, why should I be Bayesian at all? And adaptive designs are one of the key issues where, the first thing is just the whole paradigm, the idea that we collect evidence, we update, we collect more evidence, we update, we collect more evidence, we update. That looks exactly like the interims in a trial. But the other thing that goes on, which is really important, is, there's a standard example that, if you do sequential analyses in grad school, they tell you, if I collect 40 observations and I get 20 successes and I need to compute a p-value for that. If I ran an experiment where I collected 40 observations and counted the number of successes, I get 1 p-value. If I ran an experiment to collect 20 successes and I measure the number of trials, I get a different p-value. That doesn't happen with a Bayesian. And so the notion is this gives you some flexibility. So if you're a Bayesian, you can go ahead and you get the same answer regardless of how you designed the trial. And so the ability that we've had so far to use this while also maintaining type 1 error control and regulatory standards, this has been, a very simplifying assumption that has kept things interpretable and feasible.
Yeah, and it's kind of interesting, and you made a comment that I've been sort of doing this for 25 years. We've been building trials like this. I've been for 25 years. And it's amazing to think back then to now where simulation control of a trial was very rare. We actually did it even 15, 20 years ago, but it was very rare, very controversial. I think it was sort of dismissed by a lot. And now here it is, very clear, represented in a guidance document. And we've had other of these complex innovative designs, guidance documents, but very clearly that this is a scientifically acceptable thing to do, which is great to see. Okay. So the other thing that a big part of this, and the if you look at how much time and effort the guidance talks about different parts of it, it spends a lot less time on the cases where you're not borrowing data. But it says Bayesian can be used there. It can facilitate more complicated designs. I think that's all straightforward. It spends a lot more time on the borrowing part. That in this case, that Bayes equals borrowing part of thing. And if you read a lot of it, it spends a lot of time on that. And I think that's not inappropriate, that those are harder scientific cases. But it spends a lot of time. I think it's a really valuable document also for thinking about in what circumstances is borrowing reasonable, the impact it has. And this is almost bigger than the statistics part. I think this is the scientific guidance is also helpful for that idea of bringing in external data.
Yep. No, I agree completely. I do think it's important that they have said repeatedly that Bayesian is available for all designs. They talk about this in several parts that, they basically are upfront that we're emphasizing the things that we think you're going to have the most discussion on while saying you can do whatever, what you need to do to bring in external evidence. I'm hoping this will finally put to bed we occasionally get FDA doesn't take Bayesian designs. We get doesn't take adaptive. And it's sort of like, well, we've been doing this for 15 years. Are you sure? So.
Yep. Okay, a couple of the interesting maybe things it does that I maybe more statistical. It defines an analysis prior and a design prior, which I think was kind of interesting that it does. So what is an analysis prior and what's a design prior, Kurt?
So the analysis prior is the thing that's actually in the protocol. This is what drives your inferences when you say the trial will be a success if the following condition is met. That's the analysis prior. There's one of them. It says this is the one that we've decided. We've got agreement with the FDA. The sponsor's okay with it. We feel this is the best answer we can have. Right's always a loaded term for Bayesian, but it's the best answer we can get. But of course, everybody always makes the obvious point that Bayesian analyses are sensitive to the prior. If you pick a different prior, you'll get different answers. So one thing that we want to do is we want to get an idea of that sensitivity. And we want to get an idea of that sensitivity for a couple different reasons. One is when we get data at the end, we want to make sure that if we had different people, I know your advisor, Jay Cadane, has a lot of examples of a trial. You should always think of an optimist and a pessimist, and you run the trial until the optimist and the pessimist finally come into agreement, whether that's however it lands, the drug works, the drug doesn't work, whatever it may be. So you want to make sure that you've got that kind of agreement. So different design priors, I personally, you may think of them a little differently. I think of them as different people that might be analyzing the trial. The other thing that you care about here is we want to do operating characteristics. How well does the trial work? Does it answer the question? And knowing that our prior may not completely reflect reality, We want to make sure it's robust to that. So we want to make sure, hey, if our prior isn't correct, we're still going to get the right answer. We're still going to treat future patients well. We're still going to estimate parameters effectively. It talks about mean square error of estimates. But this is a robustness issue, robustness to the prior and being able to convince a broad audience. But analysis priors are what the trial actually is, how it drives decisions, and design priors are a lot of different other perspectives to make sure that it's robust and gives an answer to everybody that satisfies.
Yeah, so I could simulate the trial with the analysis prior as part of the design, part of the analysis, where a lot of times we assume that the parameter has a specific value. The probability of success is 50%, or it's 60%, or it's 70%. And we assume that. Now it's looking at bringing in also that there's a distribution to that. And especially if we're bringing in external data, we may come in and say the distribution is centered on 70%, but has this variability. How is the trial going to behave? Which now becomes a really interesting thing. And the document lays this out as different design priors, which it does give reference to skeptical and enthusiastic as you referenced. So I thought that was interesting. And that might change a little bit our behavior and what we put in or what people are asking for is that terminology, which I think was helpful.
Yeah, no, I agree. And we've done this. We've talked about if you just run a phase 2 trial, you get a broad confidence interval for the effect. You're running phase 3. If the drug truly works at the top end of that confidence interval, you need a tiny trial. If it works at the bottom end, usually the bottom end, it's not even effective. And so you want to think about, the whole distribution, the range of those values. I don't think this is going to change. You still want to make sure that individual parameters work well. The futility rules apply to part of the scale. Success rules apply more on the effective side. We're still going to look at individual points in that distribution, but the average behavior is going to be really important.
Another interesting terminology they used, and it'd be interesting to find out more about it. You and I love Bayesian hierarchical modeling, Bayesian borrowing. The term, a lot of times when you're using models like that, a Bayesian hierarchical models, we talk about borrowing. We talk about sometimes shrinkage. The FDA guidance uses the term discounting.
Yep.
Now, we very much, if I have an adult population of 1,000 patients and I have 50 pediatrics and I'm estimating pediatrics, I'm not going to lump 1,000 adults with the 50. They'd be completely overwhelmed. So most of our Bayesian techniques discount the 1,000. They borrow some level of that. They're really one and the same, but it was interesting the choice of term that the FDA refers to this as discounting rather than shrinkage or borrowing.
Yeah. So, and obviously I use the dynamic borrowing a lot in talks. I think that probably is the more common term here than discounting. I tend to think of discounting in terms of if I'm borrowing from a population and I say every patient that I'm borrowing counts the same as a fourth or a half or whatever number you agree with. Typically with FDA, what do we agree? We agree something like 1/4 to 2/3 is a common range to do here. It depends on the situation. But anyway, you basically give a weight and you say, okay, that's how much I'm going to do it. The models that we fit now, we actually allow the data to determine what that value is. And the notion is if the data agree, our current trial agrees with history, we're going to go with a higher weight. If it doesn't, then preferably we'd like that weight to be 0. And we want to say, look, this doesn't match. We're in, the different set of patients. They're behaving differently. We don't want to borrow anymore because of what we've seen. And so this is this idea of dynamic borrowing. They've related it to dynamic discounting. We've even seen this in multiple stages where people they'll pick a weight and then they'll separately do a analysis with that weight. rather than an encompassing design. But essentially, that's what it's getting on. I think we've talked about this before. CDRH has a lot more comfort with static borrowing in a lot of cases. And I think that's because devices are just different than drugs. There's less reason to think you're going to have these, it's completely different situations. But it was an interesting aspect of the guidance and emphasizes some of the differences here.
So we use the term dynamic borrowing all the time, and now the guidance refers to dynamic discounting is a term. And I think the guidance document generally gives a positive to the dynamic discounting as opposed to static. One of the things it says, dynamic approaches are used in many cases due to the more advantageous operating characteristics, such as with respect to bias and mean squared error, resulting from the lesser borrowing in cases of prior data conflict. So if it turns out that things are just looking differently, we discount them more, we borrow less. And I think it gives a nod that these are beneficial. So that was kind of interesting in the guidance.
Yep. I think we've also designed adaptive trials that have changed their sample size related to how much that prior data conflict exists, so that you can, if it doesn't look like you ought to borrow, do more in the current trial and make the trial stand on its own.
It doesn't, one of my, a Bayesian thing I love are predictive probabilities. It doesn't say predictive probabilities, but I think that's okay. I don't think we're putting predictive probabilities in labels. I don't think we're using the analysis prior, for example, and the posterior is usually driving those conclusions. They drive a lot of adaptive designs. They're used for futility. But I think it's okay that it doesn't mention them. It certainly doesn't mean they can't be used as a utility in our adaptive designs.
Well, there's going to be lots of places. I mean, we do a lot of we do longitudinal modeling in order to decide whether to stop. We know that 6-month outcomes in a knee trial are highly predictive of 2-year outcomes. We use those we have a Bayesian model relating those in our adaptive trials. But as you said, those drop out of the final analysis. And so I think they'll appear, they're implicitly mentioned when we need to think about all those priors in the guidance, but it's less of driving the main conclusion.
I was a bit surprised. There's a couple places in there where they talk about elicitation of expert opinion. And I was a little surprised it was in there. I think it's great. We have not done this much. We've not been involved in too many cases of this. But I think it opens up the door of this being a possibility. And it says, in some cases, it may be helpful to conduct a formal expert elicitation exercise with subject matter experts to incorporate the degree of consensus in the level of borrowing. That's a particular to that. So I think that that's kind of an interesting part, and it'd be interesting to see if this is something used more.
Yeah, I think there's a whole opening here on, opening's the wrong word, but basically this has opened up the elicitation and eliminating type 1 error control. This is things that the FDA hasn't done a lot of in the past. We've really been in type 1 error world for a while. And so we'll be curious, what will be the process for making that happen. A lot of stuff's been written, elicitation, we want independent experts. We want to get all ends of the spectrum, make sure you have all the views incorporated in there, but make sure they're incorporated to the degree that they represent the population. You don't want to just look at outliers, so to speak. How are we going to make sure that sponsor A and sponsor B are treated fairly when you're using informative priors? That happens in borrowing. But anyway, there's going to be a lot of work going forward in here to implement those aspects. And I hope we're a part of that.
Yeah, and it'll be interesting, a couple interesting sort of the impact of this. Let's think about the impact of this guidance document. And other times I've found myself almost defensive when the guidance document may not be overly positive. of, is this going to have an impact? And I usually say, look, nowhere does it say I can't do something, and we go forward and do it. Here's somewhat of the opposite, that this is very positive. And so the interesting thing will be the impact. Will sponsors see this and open the door to this? Open the door that the FDA accepts these, that they're open to these ideas, to it. There's also the far extreme of this. Does it bring out, I'll call it, bad actors is a hard thing, but cases where people want to do things that we think is scientifically not credible. And we do get that occasionally, that they've tried every frequentist approach and their data doesn't say success. Maybe the Bayesians will say it's successful, sort of thing. And those are not fruitful circumstances. So it'll be interesting, both the relative acceptance, does this increase Bayesian methods? And, do we have the sort of bad actors that are going to want to do inappropriate borrowing of data that's inappropriate and say, oh, you say you accept data borrowing. We should be able to use this.
Well, I mean, we know there's lots of data you shouldn't borrow from. Data that's very old, lots of data comes from different patient populations, and the guidance goes through this in a lot of detail on, you should say, no, I've been told point blank by the FDA, we're fine with the methodology. This data we don't think applies. And it's a very reasonable argument to make. As you said, the thing that terrifies me the most is people using Bayesian entirely to lower the bar. I didn't win. I got a p-value of 0.14, one-sided. Oh, if I say it's 86% superiority probability, it'll sound high and I'll get approved. That kind of stuff worries me to the ends of the earth. It's very different than the borrowing aspect, where we're trying to argue the totality of the evidence. This trial is a piece. What we're borrowing from is the other piece. We're not lowering the bar. We're putting it together. And you need people to understand that well enough that, anyway, you can see people deliberately missing the nuance, shall we say? And anyway, we've told a lot of people no who have come to us with that kind of request.
Mm-hmm. So there's a difference between lowering the bar and the totality of evidence jumping over the high bar.
Yeah,
which I think this is trying to present as these possibilities. Yeah. Absolutely.
But Scott, one more thing I was going to say. You think about the adoption curve of a new technology. We talk about early adopters and late adopters. I think that one key aspect of this is, with this guidance, if you're doing it now, you're a late adopter. It's been accepted by regulators. You're not pushing the boundaries. They've said, please, 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 Bayesian is fine. The FDA commissioner has said we view it as a step forward. So I think this has advanced the adoption curve considerably with its mere existence.
Yeah. You will still get some that say, show me 10 trials that have used this and have been successful exactly in my field, exactly the things. There will be late adopters.
We have trouble with, have you shown me 10 trials in my field and so on. If you just ask for 10 trials, we've got 3 on the first page of the guidance. So.
Yep. The other part about the impact of this, so interesting to see the impact of this of sponsors. What about the global impact? I think if you think about where the U.S. coming out with this guidance, I think it would be considered by many much more open to Bayesian techniques than the ICH E20. They're different guidances. A harmonization guidance is largely, everybody agrees with this sort of thing. And so I think they're naturally going to be behind. I think this is going to it's going to increase the use of Bayesian methods. I think it's going to show their value. I think that these are going to be more well understood by a broader audience. And I think the use is going to globally raise and make changes because of the increased use that may be driven by the U.S. Positive reaction to these. What do you think about the global scene?
Well, as you said, I don't think anybody who wasn't Bayesian yesterday, so to speak, that's not in the U.S. and not at the FDA. They're not going to, change their mind overnight. It's going to be a data point. Everybody has open minds. I do think what you said, though, is the most important part, which is getting examples, more and more examples of Bayesian things that we actually see them work. That's what convinces people. This trial, I saw what the benefit was. I saw what it does to save sample size, get cleaner solutions, better solutions in many cases, the basket trials and so on. And so I think that that's where we're really headed. We think this is the good way to go. We want to do a lot of these examples. If there are problems, we want to fix them. And we think that this is going to prove itself over time. So this is an opportunity for Bayes to prove itself. And that's great.
Yep. Agree. And it'd be interesting, I think, for example, when we look back over the years, I think the pandemic had a huge impact on platform trials because they were used, they were published, people saw them. Oh, I get it. Bayesian was used a bit in the pandemic, a good number of examples. Janssen's vaccine was approved in a Bayesian adaptive trial. And it's, I get it. This makes sense. This is reasonable. It has huge impact on then the use of it. I think this is going to be part of that, as well as signaling that. And maybe the existence of this document is in part because of COVID and other experiences and sort of seeing that. So I think this is a very positive move scientifically for sure.
And we learned a ton about platforms in doing those trials. The design that we do today is better than the design that we did in 2019 or 2020 or whenever these came out. And so, the more of these Bayesian designs we do, they'll get simpler and standardized and cleaner. That's all to the good.
Yep. So it's an exciting day with the new guidance. All of you out there, the field is open. Bring your Bayesian on. Any concluding remarks, Kurt?
I think I'm happy to see this. We've been working on this for many, many years. I applaud the FDA folks that it's a long document. It's got a lot of content in it. There was a lot of work going in on all the reviews that led to this. So we appreciate their time. We appreciate everybody who spends time on these kind of guidance documents and how much effort and thoroughness is needed to do them right. So that's my concluding remark.
Yeah, a very well written document, a great effort. We're excited about it. All right, thank you all for joining us here in the interim. Until the next time, we will be here in the interim.